This conference will now be recorded.
Hello everyone. In biomolecule chapter, carbohydrate, which is one of the important biomolecule, that part is done. Next important biomolecule is protein, and related to protein there will be some other topics, that is amino acid, which is basically the building block of protein. Amino acid, its structure, properties, then stereochemistry, what are the symbols we used for amino acid, then peptide bond formation between amino acid and different types of peptides. All this we'll discuss today. So what is protein, which is the actual biomolecule, and amino acid, it is the basic unit of protein. So proteins are the most abundant biomolecule if we consider in all living organisms. So the term protein, how it is derived? It is derived from the Greek word proteose. Okay, this means primary or holding first place. They are the main functional unit for the living things. Now, what are their, that is, importance? Why we are discussing it? Because they are involved in every function of the cell, including, for example, respiration. And amino acid, basically alpha amino acid, not beta or gamma. There are different types of amino acid, but only alpha amino acid, they are actually the monomer of protein, because protein itself is a very large molecule. If you consider protein as polymer, then the basic unit, that is the repeating unit, that will be alpha amino acid. So first we have to start from the basic unit, which is alpha amino acid. Then only we can understand the detailed structure and roles of protein. First, if we directly start from protein, it is not possible, because if we do not have proper idea about the basic unit, then protein, which is very large biomolecule, it will not be possible for us to understand its structure. Okay, so that is why first we have to know what is this monomer, basic unit, which is alpha amino acid. And once we know the alpha amino acid, its structure, properties, stereochemistry, then we will be able to understand how small alpha amino acid molecules, they are connected to each other, and now they are forming the large biomolecule, which is protein. So alpha amino acid, simply we call it amino acid. So when we are saying amino acid in protein structure, obviously it means alpha amino acid. Now if we consider these two terms individually, amino and acid, amine group is NH2, right? And acid is CO2H. That means these two groups will be present in the molecule. So that is the reason why the name is amino acid, carboxylic acid group and amino. Now protein molecules are made up of this alpha amino acid. Now why we are calling it alpha, why not beta or gamma? Because the carbon with which NH2 group is attached, in the same molecule CO2H group is also attached. So that is the reason it is alpha amino acid. And as carbon tetravalency we have to fulfill, there will be one hydrogen atom and another group will be there. Okay, so we have this structure here. So here in the same carbon, both these groups are attached. So that is why it is called alpha amino acid. Suppose if they are attached to different carbon, if the structure is like this, two carbon, one carbon there is NH2 group, another carbon there is CO2H group. So in that case you can call it beta, or if it is 1,3 carbon, then it will be gamma. So in this way, depending on the position of NH2 and CO2H group, different types of amino acid. But in this case, we are interested only in alpha amino acid, not beta or gamma, because on the alpha amino acid is the amino acid protein molecules are made of. There are 20 alpha amino acids that are commonly found in protein molecules. So we have to know only these 20 alpha amino acids. And each amino acid, there is a trivial name. Now this trivial name, it may be two types. One is 3-letter code or another one is 1-letter code. So those names we will see. Some you can also call it symbols for alpha-amino acid after we see the detailed structure of the amino acid. Then we'll go to the name. But remember, we use either 3-letter code or 1-letter code for alpha-amino acid. So alpha-amino acid, carbon, why star mark is given? Because why star mark is given? Because if R group is not H, under that condition, all the 4 groups are different, so this carbon is always chiral. Only if it is H, then 2 groups are same, that is 2 hydrogen, same atom. So now the carbon is not chiral. But except this only one case where R is equal to H. In all other cases, all the 4 groups are different, so it will be chiral carbon. So this is the basic alpha amino acid structure, and here these 3 parts, it will not be changed. Only the part R, that will be changed. Okay, so depending on this nature of R group, we can have different alpha amino acid. Structure of alpha amino acid, we have amino group, carboxyl group, and this R group, it is called side chain. So there are actually thousands of amino acids that can occur in nature, but we are interested only in 20 alpha amino acid that will form a part of the protein in the human body. Okay, only these 20, not any other amino acid. Although all these have varied structure, but the basic structure remains uniform. So you can see in the right hand side, the basic structure, there will be one amino group that is fixed. There will be one CO2H group that is also fixed. H is fixed. Only the side chain R will vary. Now, though I am saying here that amine group, NH2 group is fixed, but there is only one exception. We'll discuss that in detail when we'll see the structure of that particular amino acid. But except that, in all cases, you will find this primary NH2 amine group, that is NH2, and CO2H, H. These 3 fixed. Only these are side chain that will vary, and that is the reason we can have different amino acid. So this carbon is alpha carbon, and in the same carbon both the NH2 and CO2H groups are attached. This atom is surrounded by 3 chemical groups, NH2, CO2H, then the third group, it is denoted by R, and that is also known as side chain, and it will be different for different amino acid, and that will be the reason for its uniqueness. Now, though you are seeing the first structure where NH2 group is written as neutral NH2, CO2H group is written as neutral CO2H group, but actually what happens, amino acid, they remain, look at the second structure, they remain like this. So here what happens, as here basic group, this is basic group, and this group is acidic. So here both acidic and basic groups are present simultaneously in the same molecule. So that is the reason here transfer of hydrogen, transfer of this hydrogen is possible towards this side. So as if it is intramolecular proton transfer from CO2H towards NH2 group. So that is the reason now, because of this extra hydrogen, NH2 is now NH3, and nitrogen, because of 4 bonds, it will be now positively charged because one extra H plus is now attached, and H is removed from CO2H, and that is the reason negative charge will be there. Now this type of ion is known as Zwitter ion. Zwitter ion means where both positive charge as well as negative charge will remain simultaneously in a single molecule. It is not that positive charge on one molecule, negative charge on other molecule. No, it is transfer within the same molecule, transfer of proton, and positive charge is present, one negative charge is present, but overall the molecule is neutral because single positive charge balanced by single negative charge. Okay, so the molecule is neutral, and this is known as Zwitter ion. Alpha amino acid, they mostly remain in this form, not the form that you are seeing. That is the first picture. Classification of alpha amino acid. So 20 alpha amino acid that are commonly found in the protein molecules. we already know that three things are fixed, three groups, NH2, H, CO2H. Only the R group will change. Now this R group may be different types. It may be acidic group, it may be basic, it may be polar, it may be non-polar. So depending on that, we can have some classification for amino acid. Okay, so one by one we will see. The first type that is possible, here it is written non-polar and aliphatic R group. So aliphatic means no aromatic ring will be there, no benzene ring, and it will be non-polar. Now when we say non-polar, mostly it will be hydrocarbon, that is carbon and hydrogen. These two atoms will be present only. So the first alpha amino acid that you are seeing, which is glycine, this is the simplest structure possible because here the R group, which is in blue colored region, here R group simplest, that is hydrogen atom. So this is known as glycine, and this is the only alpha amino acid which is not chiral, that is alpha carbon. If you see two same atom, so two groups are same, obviously it will not be chiral. But in other cases, when the group R is not H, look at the next molecule, CH3, name is alanine. So when it is CH3, now it is chiral, but important thing is the groups are non-polar and aliphatic. So it is hydrocarbon CH3. Then the next molecule, gradually the R group, it is becoming more complex. It is valine and the group is isopropyl. CH3 whole to CH. This part. Rest of the part is same for all the molecules. Only you have to focus on the blue box where the R group is highlighted. Leucine. Then one more carbon, but only carbon and hydrogen. There is no no heteroatom, oxygen, nitrogen, nothing. Then the next one, now we have one sulfur, but still it is non-polar. Though it is not purely hydrocarbon, but still it is non-polar. Okay, because both sides of sulfur we have alkyl group, so there will be no polarity generation. This is known as methionine. Then the next molecule, this is also a hydrocarbon chain, total 4 carbon. This is also 4 carbon. Leucine is also 4 carbon. Only difference is that as if this methyl group is now attached to this carbon. That is the only difference. Otherwise it is same. So the name is also, it pronunciation, you can find some similarity, leucine and isoleucine. So this is the type where R group is non-polar, mostly hydrocarbon, and it is aliphatic. Next possibility, polar group. Polar and uncharged R group. So here the R group, there will be no charge present and it should be polar. So the first one is CH2H. Now OH group, because of this particular part, this will be as if it is alcohol, right? OH group. So it is serine. Then CHOH, as if one hydrogen is replaced by methyl group, name is threonine. Then CH2SH. Now see, in the previous category, methionine, there also you have seen sulfur, but both side we have alkyl group. One side there is 2 carbon, another side 1 carbon. But in this case, it is as if analog of alcohol. Just in place of O, oxygen, if you write S, it will be SH. So it is analog of alcohol. And one side there is alkyl group, another side there is H. So now it will be having some polarity. Okay, both sides not similar type of groups. One side H, another side hydrocarbon. So cysteine, it will be under polar category. Then we have proline. Now this is having a very unique structure where you will not find NH2, that is primary amine, you will not find in this case. This is primary amine, right? 1D. But if you look at this structure, here the NH group, it is like this. H, CO2H, and there is some ring structure. That means as if here this is the R group, but the R group, it is attached with NH. Fine. So the structure is like this. NH. So including the carbon and nitrogen, it is 5-membered ring. 1, 2, 3, 4, 5. Okay. I'm numbering it here 1, 2, 3, 4, 5, like this 1, 2, 3. Okay, so this is the only alpha amino acid where the NH is not primary, it is actually secondary. Only one H is present, both sides there is some group, it is actually ring structure. And if you consider deuteron, obviously this H will be transferred here. So now it is NH2 plus NH2, one proton extra, so that is why there will be positive charge. So this is proline. The next one is CH2 CONH2. So here we have amide group CONH2 and also another CH2. So this blue part, if you see, it is CH2 CONH2. Name is asparagine. Then glutamine. Glutamine is also just like asparagine, only difference is one carbon extra. Otherwise, there is no difference. So this category, second one, polar uncharged earth group. So in these two category, how many amino acids we have seen? Total 6 here, and here also it is total 6. So 12 alpha amino acid we have seen, 8 more we have to see. Positively charged R group. Now there will be some charge present in the R group, and one interesting fact is that all these three examples that you are seeing here, the side chain is basic in nature. Okay, so see the side chain 4 carbon and then there is NH2, but it is in protonated form, so that is why we are saying positively charged. Now as this alpha amino acid mostly they are in vivo, that is inside the body, and if you consider the pH, so pH is such they remain in charged condition. Sometimes in some places you can see example of alpha amino acid where lysine, maybe it will be, you can see that it is written as CH2 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 4 times, that is 4 carbon chain, linear chain, and then there is NH2, but actually it remains under normal condition in vivo, it remains in this protonated form, so that is why it is positively charged, NH3 plus. And this part there is no change, it is just the deuteron structure we have already seen. So lysine, it is actually basic and one proton is extra attached, so that is why it is positively charged. Similarly, if you consider arginine, after 3 carbon linear chain there is NH, then C double bond NH. If you do not consider this extra proton, if you consider it, it will be positively charged. But as if it is like this, if I write it 3 carbon, it is starting here 1, 2, 3. So 3 CH2, and after that it is NH, C double bond NH, then NH2. So this is actually the side chain. But if I am adding extra proton here, now it will be positive. Okay. So but remember, this is, you can also say example of basic amino acid because the side chain is containing this type of amine. Now the third one, histidine. Here we have CH2, and after that if I write it separately below, it is like this, as if CH2 and then there is 5-membered ring attached, and in this 5-membered ring there is carbon. So if I am numbering it like this, 1, 2, 3, 4, 5. So it is 1, at 2 position there is NH. First I am writing neutral NH, then CH which is third position, then double bond, and we have N here, and then CH. It is actually CH2. Fine. So if you consider this structure, it will be double bond. So if you consider this structure, it is 4. It is 4. It is 5. Here, if we add one extra hydrogen in this nitrogen, then it will be positively charged. So this is the structure of histidine, which is very unique because in this case you will find a 5-member ring, and the 5-member ring, it is containing 2 nitrogen and 3 carbon. There is a 2. Fine. So this is also basic. because of presence of NH. The next category, negatively charged R group. You can also say this is acidic side chain. And the previous one you can say this is basic side chain. So acidic side chain, now it will be in this form, that is one H is removed. It is you can also write it as CH2CO2H, and in that case the name will be aspartic acid. But if you are writing in the deprotonated form, that is CH2CO2 minus, name will be different because now it is anion. One extra negative charge is there. So this positive charge is balanced by this, but still there is one negative charge extra. So overall there will be one extra negative charge. So that is why it is under this category, and the name is aspartate. But if you are writing CO2H, then you should call it aspartic acid. Similarly, the next structure, it is completely same, only one CH2 is extra, and if you are writing it with negative charge, it will be glutamate. Otherwise you can also call it glutamic acid, but in that case you have to write it as CO2H. And the last category, again non-polar. Remember the first category, non-polar aliphatic. This is also non-polar, but now it is aromatic. So carbon, hydrogen mostly will be present, but there will be aromatic ring. So the first one is known as phenylalanine. Remember, when it is alanine amino acid, the side chain is methyl group, but when it is phenylalanine, as if one H of carbon is replaced by pH. So now it is CH2PH, and this is phenylalanine, as if derivative of alanine. Then tyrosine, just like phenylalanine, only difference this position, now there is OH group, that is the only difference. And then we have tryptophan, where CH2 group is there, and then there is a heterocycle, and this molecule, that is the circled part, this is actually known as indole. So indole is this structure. Which is important heterocycle, and this heterocycle moiety, now it is present in the side chain along with this CH2. So CH2, then indole, as if this is the connection point where it is attached to this CH2. But considering this CH2 and the indole moiety together, it is the amino acid which is known as tryptophan. Fine. Now structure, why it is important to remember, because if you, when we'll discuss question, you will be getting the idea that sometimes the symbols of amino acid will be given. Those symbols we have not yet discussed, but today's class only we will discuss it. So when symbols will be given in the problem, then from the symbol you have to recognize which amino acid the problem is talking about. So that is why remembering the structure along with the name is very important. Fine. So now we will see the symbols. There will be 3-letter symbol and also 1-letter symbol. So some amino acid in the left hand side, some are in the right hand side. Alanine, it is ALA, that is the 3-letter symbol. Mostly the problems that you will get, you will see the use of 3-letter symbol, not a single letter. Okay. So arginine, ARG, mostly those letters when we pronounce, only those letters where there is intense pronunciation is there, those letters are used. So see, when it is asparagine, A, S and N, these 3 are considered. Then aspartic acid, the first 3 letters are considered. Cysteine, first 3 letters. Then glutamic acid, you can also say glutamate, depending on you are writing it as CO2H minus, CO2H or CO2 minus. It may be glutamic acid or glutamate, whatever it is, but it is GLU. And single letter, what is the exact reason why it is used? Suppose why D for aspartic acid? There is no hard and fast rule, you just have to remember, but mostly 3-letter symbols will be used, that is will be given in the problem. Then glutamine, which is amide, GLN. Then glycine, the first 3 letters. Histidine, again first 3. Isoleucine, I, L and then E. leucine, first 3. Lysine, first 3. Then methionine, first 3. Same. So in this way, it will be easier to remember in all these cases. Now TRP, not Y. Then tyrosine, first 3. Valine, first 3. Fine. Now here we have total 20 amino acid, as we know. But remember, among these 20 amino acid, some amino acids can be synthesized by us in vivo. So that is why they will be non-essential amino acid. But there are some amino acid that needs to be obtained through diet because we cannot synthesize them. So that is why it is very essential that we take it through diet. So they are called as essential amino acid. So here the amino acid that are marked, highlighted with pink box, they are actually the essential amino acid. Fine. 10 essential amino acids are there out of 20. Properties of amino acid. So these amino acids, as we have seen, there is positive charge and negative charge present simultaneously. So it is basically, it will behave like ionic compounds, and it will be mostly crystalline solid and also water soluble. Normally when we consider any organic molecule, they are not water soluble, right? But because of the presence of this type of charge that we have seen in Zwitter ion form, it will be behaving as if it is just like inorganic salt. Okay, so this is very unique, though they are organic molecule, but still their behavior is different. Okay, so colorless, water soluble, crystalline solid, insoluble in non-polar solvents. See, all these organic solvent if you use, it is not soluble. Now since they have both CO2H group as well as NH2 group simultaneously, don't think that their properties will be same as regular amine and carboxylic acid. Suppose if you are considering any amine having structure RNH2, or if you are considering any carboxylic acid structure, that is R CO2H. So individually I am writing two different molecules. But when both these groups are present simultaneously in the same molecule, obviously you cannot expect the properties that you expect from single NH2 containing compound or single CO2H containing. So now it will be completely different. Their properties differ from regular amine and carboxylic acid. Fine, because they are present together in the same molecule. Now as there are two different types of groups, one is basic, another one is acidic. So what happens, there is a specific pH that is a negative logarithm of H plus concentration. pH means negative logarithm of H plus concentration. So as there is a negative sign, when H plus concentration is very high, that means it is acidic. Then the value of pH will be very low because there is a negative sign. Similarly, if H plus concentration is very low, that means log H plus is low, and there will be negative will be positive, and you will see the value is very high. So when value of pH is high, mostly it is considered that 7 is the middle point. If it is lesser than 7, it is towards acidic side. If it is greater than 7, it is towards basic side. Fine. So these two are inversely related, that is H plus concentration and pH inversely related. When acidity is very high, pH will be low, and vice versa. Okay. So there is a specific pH for each alpha amino acid where the net charge of an amine group and the acid, that is the net charge of the amino acid molecule, that will be neutral. So at that particular pH, not just any pH, at that particular pH, there will be positive charge over NH2, that is now it will be NH3 plus and CO2 minus. That means overall it will be neutral, and it will show amphoteric behavior. It will not show acidic property or basic property because plus is compensated by the minus. Fine. So this particular pH is called isoelectric point, PI, and don't think that this pH is same for all the amino acid. That is not possible because R groups are not same. So for each amino acid there is specific, that is different value of isoelectric point. Fine. So here, if you look at this, the middle form, this is the general structure alpha, because we have not mentioned what is the nature of R form. We have written the general structure. In this general structure, when it is neutral pH, now not necessary it will be, again I am saying that isoelectric point it will be same for all amino acid, but as we are discussing with the general structure, we are keeping the discussion for general structure only. So that is why it is written neutral pH, but it may be little bit plus minus if you change to some other amino acid. Okay, that is for different amino acid, it may be slightly different. So Zuitara neutral pH, net charge is 0 because the single positive charge is balanced by negative charge. Now if you are increasing HO minus concentration, that means if you are going towards basic condition, so pH is high, alkaline pH means high pH compared to this neutral pH. Now what happens, because of this HO minus, presence of HO minus, it will capture this proton from NH3, and as a result now this part is neutral, so you have single negative charge present here, so net charge is -1. And if you move towards right hand side, follow the forward arrow, H plus is now added. That means now we are moving towards acidic pH. And when H plus is added, obviously this CO2 negative, now it will be capturing this H plus, it is now neutral. So there is extra positive charge, so net charge is +1. Now among these three forms, suppose this is form number 1, this is 2, this is 3. Among these three forms, which form will be predominating? Predominant form of amino acid depends on the pH of the solution. So for any specific amino acid, there is some fixed value for isoelectric point, that is a fixed pH, and if the pH is greater than that, it will remain in the form number 3, but if it is lesser than that particular pH value, then it will be having in form number 1. And if you can maintain that particular isoelectric point for that specific alpha amino acid, then it will remain in form number 2. Okay, and this isoelectric point, that will be different for different amino acid depending on the R. Fine. So predominant form of amino acid depends on the pH of the solution obviously, and also on the nature of the R group, what type of R group we have taken. Fine. So there is a problem, the correct structure of histidine. So here full name is given, no trivial name is there, and it is asking the correct structure of histidine in strongly acidic solution. And what is the pH? Strongly acidic, pH is given 2. So first we have to know what is the normal structure of histidine, right? So histidine, the general form you can easily write. So NH3 plus will be there, then 1 H will be there, 1 CO2 minus will be there, and the R group for histidine is CH2, and then there is a 5-member ring, and in this 5-member ring there is NH, then double bond, and also N. So this is the neutral form I have written for the R group. This is the actual structure of histidine. But as pH is low, so what will happen? There will be excess proton. It is acidic. So under acidic condition, here lone pair that is present over this nitrogen, that will be captured by, that is this lone pair will be taken by H. So as a result, this nitrogen, it will be now NH, and there will be positive charge. Now if you are thinking why not the other nitrogen is capturing the H plus, that is why not this one. So what happens if you consider this nitrogen, here lone pair availability and the lone pair which is present here, suppose this is nitrogen number 2, this is nitrogen number 1. If you compare lone pair availability over nitrogen number 1 and nitrogen number 2, lone pair availability is more in case of nitrogen number 2. Why? Because in case of nitrogen number 1, this type of resonance is possible. So after resonance, the structure will be like this. So there is negative charge over nitrogen. Fine. This is positive. So that is the reason lone pair availability will be less because of this type of resonance, but resonance is not possible in case of 2. So lone pair availability is high. So proton should be taken by this lone pair only, not the nitrogen number 1. So that is the reason. Though it is true that there are 2 basic sites, but when it is acidic medium, you can consider this NH2, it is already in zwitter ion form with this CO2H. Okay, so there is positive charge and negative charge. And when it is in acidic medium, then what will happen? If you, okay, 2 way you can consider. Suppose if you are not writing the zwitter ion structure, then it is like this: NH2 simple neutral, and this is CO2H. And there is no change in CO2H because it is already acidic, no effect in acidic medium. But these 2 basic sites, they will be protonated. So if you consider this logic, this is the structure, and according to that, the correct option will be option 4, right? You can also consider another point of view. Suppose initially this CO2H, it is present as COO minus initially. That is, I am starting with zwitter ion. And NH2 is present as NH3 plus. Now under this condition, if you are increasing, that is if you are maintaining pH 2, what will happen? This NH3 is already positive. It cannot accept one more. It is not possible to accept again H plus. But CO2 is minus, so it can capture H plus. And this part also there will be proton attachment. So as a result, again you are reaching the same conclusion. So whatever is the starting point, whether you start from neutral group as it is mentioned in this picture, or if you start from zwitter ion form, you will reach the same answer. Okay, so option 4 is the correct option. If you look at the other options, obviously this is not possible because CO2, it will not remain in negative form. Yes, it is so acidic pH, right? So obviously it will capture it. So this cannot be correct. Then option 1, this is not at all possible because when it is CO2H, how there will be negative charge? It is not possible at all. You can easily cancel it. And the option 3. In option 3, here what happens? As if here nitrogen number 1 is protonated. So see, this nitrogen is still neutral. This is nitrogen 2, this is 1. But as I said, because of resonance, lone pair ability over 2 nitrogen is more. So these should be protonated, not the other one. Otherwise, this option there is no problem. In this part there is no problem. Problem is only in this ring structure. If you compare these two, you will see protonation is here at nitrogen number 2, but protonation in case of option 3 it is at nitrogen number 1. So this is correct, option 4, not option 3. Fine. So though we have seen the structure of amino acid, and detailed stereochemistry obviously will not discuss, that is how to decide R and S, because that is not included in syllabus. But some primary idea about the stereochemistry of amino acid we will see here. So except glycine, you know, in case of glycine, R group is H, and here chiral it is not possible. So except glycine, all other amino acid, they are having at least one chiral carbon. That is the minimum criteria. Okay, so they will always be optically active. So at least one. There may be more than that, depending on what type of R group you have taken. But at least one is always there. Now they remain in 2 forms, mainly D and L. Now how to know D and L? This D and L designation refers to configuration of the alpha carbon regardless of the number of chiral carbon in the molecule. So as I have said that there is at least one chiral carbon, which is the alpha carbon. Alpha carbon means the carbon with which NH2 group as well as CO2H group both are attached. I am talking about that carbon. So that carbon, based on that, this D and L we can decide, not other chiral carbon, even if it is present. Now, if you look at this picture, here it is D amino acid. So when you are writing the amino acid molecule in this format. In this format means you have to keep CO2H group at upper position, R group in this lower position. Okay, under that condition, if NH2 is in the right hand side, then it will be D-amino acid. If it is left, it is L-amino acid. So it is not that you are writing CO2H below and R is up, and then you are deciding D or L. No, that is not correct way. You have to write first CO2H group in the upper part and then R in the lower part. Under that condition, if NH2 in the right hand side, then it is D. Now among these two forms, D and L, this is the general picture because we have not written what is the exact nature of R. L-amino acid are used predominantly by the living organism for synthesizing proteins. So naturally, this L form is mostly present. But it is not that D-amino acid is not present, but that is very rare. It is rarely observed in certain organisms. So this is the general picture. L form is predominant in nature. But if you see the example alanine, so R group is below, CO2H group up, and under that condition NH2 is in the right hand side, so it is D-alanine, and its other enantiomer, because these two, as there is only one chiral center, one will be R, another one will be S. So this is D and this is L. So L form is the natural form. Peptide bond formation. So now we know what is amino acid, its properties, structure, stereochemistry. We have also seen all the examples with detailed structure. Now once we know the amino acid properly, next we have to know peptide bond. So peptide bond is basically, now we will connect two individual amino acid through peptide bond. So the carboxyl group of the first amino acid, suppose you have taken two amino acids. The first amino acid, you are considering the CO2H group. It is reacting with the amine group of the second amino acid. And when there is reaction between CO2H and NH2, there is formation of amide. So amide is CONH2 group. I am writing in this way. Suppose both sides there is R. If it is CONH2, it is primary amide. If I am writing in this way, CONH, and there is some other group attached to N, then it is secondary. So if suppose it is like this, that is RCOOH. Just take any normal carboxylic acid, not necessarily it has to be alpha-amino acid. Another molecule is containing RNH2. So under this condition, if you remove this OH and one H from here, what you will get? You will get CONH, right? As if one water molecule is removed. So this is the peptide bond. Now, this is, suppose the RCO2H, in this R part there is also NH2. So this is the first amino acid. And the second amino acid, here we have written NH2 group, but in R part there is also CO2H, right? So this amide linkage will be called peptide bond, and the resulting compound will be called dipeptide. Why dipeptide? Because you have taken two amino acids. So when it is dipeptide, there is actually one peptide bond. If you see this structure, this is amino acid number 1, where R group is R1. The second amino acid, R group is R2. And how we have written this amino acid, look at it carefully. Amino acid we have written, the first one, keeping this N part in the left hand side. So this is left hand side, and CO2H group we have written in the right hand side. The same pattern we are again following for amino acid number 2, but in the left hand side we have this amine part, and the right hand side we have this CO2H. So under this condition, if there is removal of water molecule, we are getting this type of peptide bond, right? So this terminal is called N terminal and the other terminal is called C terminal. Okay, so this is the general convention which is followed. N is written first, that is in the left hand side, and C is written in the right hand side. So in this case, we have taken two amino acid. There is generation of one peptide bond. So remember, when there are two amino acid, only one peptide bond will be generated, but this will be called dipeptide. Similarly, you can also add another amino acid. Suppose this is dipeptide and now you are adding another amino acid like this. Suppose now it is R3H CO2 minus. So again there will be removal of H and OH, that is removal of water, you will get another peptide bond. So now there are total 3 amino acids. But how many peptide bond? Only 2. So addition of another amino acid to this dipeptide, it will result a second peptide bond and you will call it tripeptide. And in this way tetrapeptide, pentapeptide you can generate. So in this pentapeptide, total 5 amino acids are involved. But how many peptide bond? Only 4. So one peptide bond, total number of peptide bond, it will always be lesser than 1. That is, compared to the number of amino acids, it will always be 1 less. Each amino acid unit in peptide, it is also known as residue. That is individual. If it is pentapeptide, if I am asking that how many residues are present, then it will be, the answer is 5 residues are present. Depending on the number of this residue, different types of peptides are possible. So now once we know what is peptide bond, what are the different types of peptide possible, that we'll see. So here you can see total how many peptide bonds are there. This is one peptide bond. This is another peptide bond. Third peptide bond. So total 3 peptide bonds are there, right? But how many amino acids? Just look at this R group. Total 4 R groups. That means 4 amino acids are present. So this is basically tetrapeptide. But peptide bond total 3. And see the N terminus in the left hand side in blue color and the C terminus that is in the right hand side in pink. Fine. So this is the general convention we follow when we write any peptide. N is first, that is in the left. C, it is in the right hand side. Now once we know this general idea clear, how to write peptide, whether it is tri, penta, tetra, whatever it is. So here we have the first type. This is the simplest possible because at least 2 amino acid you have to take. So here we have taken 2 amino acid. And the 2 amino acid, one is in black color, another one it is in red color. Both the amino acid are same. Both are basically glycine. So it is how it is written. If you are writing the structure, obviously you have to write it in this way, where N terminal it is in the left, CO2H that is in the right. But when you will write the 3-letter word, remember the symbols, then you have to write it in this way, GLY-GLY. If you are using single letter, another way by which we can write the symbols, then it will be capital G, capital G. So these consist of 2 amino acid residues, and these 2 amino acid are 2 glycine amino acid. Tripeptide. So in this case, the first amino acid it is in black color, then the next one is blue color, and the last one is red color. Here stereochemistry is also given, that is the bonds above the pen, below the pen, but just ignore that part. Focus on the what are the different types of amino acid we have taken. So the first amino acid is VAL. VAL, single letter if you are writing, it will be V. Then ALA, then hyphen GLY. So the black one is basically valine, and valine R group is Me2CH. So see, 2 methyl group and then CH. Then for alanine, here the R group is methyl, simply methyl. And for GLY, the R group is H. So these are the 3 R groups present. So if you are expressing it in 3 letter, it will be val hyphen ala hyphen gly. If you are writing in 1 letter, it will be VAG. So there are total how many peptide bonds are there? This is one peptide bond. This is another. So total 2 peptide bonds are there. So when there are 3 amino acid, I am writing capital A, amino acid they are also written in this way. Then there will be 2 peptide bond. Okay. Next possibility, Now directly we are going to oligopeptide, that is tetra, penta, that is also possible, but individual names are not given. Now once it is crossing the value 3, if it is more than 3, then we will call it oligopeptide. So when there are total 4 to 10 amino acid residues, we consider them as oligopeptide. But if it is more than that, that is consists of more than 10 and less than around 50, then it will be called polypeptide. Fine. So remember, oligo is up to 10. If it is beyond 10 but lesser than around 50, because next we will see what is protein, then you will understand difference between protein and polypeptide, consists of more than 10. Fine. So here you can see all these individual amino acid, how they are written with the 3-letter symbol GLY, ILE, BALC. So this terminal is N terminal left hand side and this terminal is R terminal right hand side. And there is some SS linkage also. Remember in cysteine, if you consider the side chain, that is R group, there is SH group present. So as if there are two SH group. So these two SH group, if you are removing these two H, there will be SS linkage. So that is why these two systems, they are connected by this SS linkage, and that is the reason now there is some folding type of nature. Otherwise it can be written like this linear. But because of this SS linkage, which is also known as disulfide linkage, now it is a coil type of shape is there. Good. So types of peptides: dipeptide, tripeptide, oligopeptide, and polypeptide. These are the different types of peptide. Fine. Here we have this problem. The peptide that gives positive steric ammonium nitrate test. Steric ammonium nitrate test, in short, it is also called CAN test. C for steric, A for ammonium, N for nitrate. So in alcohol chapter, we have discussed in detail what is steric ammonium nitrate test. This test is basically performed to identify whether there is any OH group present or not. That is for the identification of alcohol. So that means if the peptide is giving positive CAN test, there must be presence of OH. And this peptide is also giving positive test of carbylamine. Now carbylamine test is possible only, it is positive only when there is primary amine group present. So this is giving positive test simultaneously for CAN as well as carbylamine. That means OH group and NH2 group both must be present. Okay. So once we know this, if you look at this peptide, in all these options, 2 amino acids are there. So GLN, it is actually that amino acid where the R group is CH2, CH2, CONH2, it is amide. And ASP, it is aspartic acid, but the R group is CH2, COOH. So these two are the side chains. I am writing only the side chain for simplicity. Then again ASP and GLN. Now GLN, it is, we have already seen the structure. It is actually same. Here N terminal GLN written first, then ASP. But in the second option, ASP is written first. That means N terminal amino acid should be ASP amino acid. The side chain is CH2CO2H. And then the next one is serine, ACR. Now in serine, the side chain is CH2OH. That means OH group is present. So these should give positive CAN test. And in lysine, the side chain is CH2 4 times. I'm writing it in this way. Then NH2. That means primary amine group present. So this is the only option where you are getting OH and NH2. Because if you look at the last option, LYC, lysine, NH2 is present, but ASP, which is having the side chain CH2CO2H. So it is not having any OH, but both groups must be present. So the condition is fulfilled only by option 3. So that is why it should be the correct option. So OH group is giving positive steric ammonium nitrate test. NH2 group, that is 1 degree amine, is giving positive carbylamine test. Okay. So that is the reason Here, this is molecule serine, this is the side chain, and lysine, this is the side chain, 4CH2 and then NH2. Now, serine will be in the N terminal. So that means the CO2H group of serine is involved in peptide bond. So see, this is the newly generated peptide bond, where in the left hand side we have serine, right hand side we have lysine. So lysine, CO2H, that is the C terminal, this is the N terminal. So because of presence of this OH group and NH2, this should be giving positive carbylamine test as well as CAN test. Now if you are thinking that in GLN and ASP also, this NH2 is always present, right? But remember, even if you consider this NH2, which is present for option 1 as well as option 2, but the problem is here OH is not present. That is possible only with serine. So that is why option 3 should be the correct option. Next question. The number of chiral carbon, or it may be more than that, plural is also possible. This is integer type of question. Present in the peptide. What is the peptide? See, total 3 amino acids are present. It is tripeptide. ILE, ARG, arginine, and PRO, that is proline. So when it is ILE, let me write the side chain. It is CH, then methyl group. So from this problem you can understand why the remembering the structure of amino acids are so much important. Then when it is ARG, now the R group is CH2 3 times, then NH, double bond NH, NH2. And when it is proline, it is actually second, sorry, let me write it, is there is presence of 2 degree M. We know proline, its structure is very unique. It is not other like other amino acid. There is 5-member ring like this. So this is secondary. But we have to find chiral carbon. So individually these are the side chain, right? But if you look at the, that is how the, what is the pattern? ILE written first. That means you have to start with isoleucine, where this is the side chain. So when you are writing isoleucine first, N terminal should be free for isoleucine. Then there is the next amino acid, which is arginine. This is the side chain, and the CO2H group of arginine, now it will be forming peptide with the nitrogen of proline. So the structure as a whole, it will be like this. So here individually, if I highlight, this is N terminal, right? According the general convention is followed, it should be in the left. So this part, this is one peptide bond, this is another peptide. But important thing is, I will separate the 3 amino acid. So this is actually ILE, isoleucine. How many chiral centers are there? This is one chiral center, okay, and another chiral center that is present in the alpha carbon. So this is the alpha carbon, so 2 chiral center. Then there is arginine. In arginine, this is the alpha carbon, okay, and this is the R group. 3 times CHO, this is the R group. In yellow color I am highlighting. So this part is ARG. Then there is peptide, and this is proline. And in proline, this is the carbon, alpha carbon, where N and CO2H groups are present. So this is also carbon. So how many chiral centers are there? 1, 2, 3, 4. So the answer should be 4. Okay. So in both the questions, if we do not know previous, we do not have previous idea about the structure of amino acid, we will not be able to answer. So remember, you have to remember all the structures. Okay. The total number of negative charge. Sorry, we are at the end of the time. So we'll finish this problem in the next class. We are ending the session here. Thank you for listening.